Bed bug, Wikipedia article audio. Bed bugs are parasitic insects of the Cimicid family that feed exclusively on blood. Cimex lectularius, the common bed bug, is the best known as it prefers to feed on human blood. Other Cimex species specialize in other animals, e.g., bat bugs, such as Cimex pipistrelli, Cimex pilocellus, and Cimex adjunctus. The name bed bug derives from the preferred habitat of Cimex lectularius, warm houses and especially near or inside beds and bedding or other sleep areas. Bed bugs are mainly active at night, but are not exclusively nocturnal. They usually feed on their hosts without being noticed. Infestation. Description. A number of adverse health effects may result from bed bug bites, including skin rashes, psychological effects, and allergic symptoms. Bed bugs are not known to transmit any pathogens as disease vectors. Certain signs and symptoms suggest the presence of bed bugs. Finding the adult insects confirms the diagnosis. Bed bugs have been known as human parasites for thousands of years. At a point in the early 1940s, they were mostly eradicated in the developed world, but have increased in prevalence since 1995, likely due to pesticide resistance, governmental bans on effective pesticides, and international travel. Because infestation of human habitats has begun to increase, bed bug bites and related conditions have also been on the rise. Diagnosis of an infestation involves both finding bed bugs and the occurrence of compatible symptoms. Treatment involves the elimination of the insect and taking measures to treat symptoms until they resolve. Bed bug bites or cimicoses may lead to a range of skin manifestations from no visible effects to prominent blisters. Effects include skin rashes, psychological effects, and allergic symptoms. Although bed bugs can be infected with at least 28 human pathogens, no studies have found that the insects are capable of transmitting any of these to humans. They have been found with methicillin-resistant Staphylococcus aureus and with vancomycin-resistant Enterococcus faecium, but the significance of this is still unknown. Physical. Investigations into potential transmission of HIV, MRSA, hepatitis B, hepatitis C, and hepatitis E have not found any evidence that bed bugs can spread these diseases. However, arboviruses may be transmissible. Adult bed bugs are light brown to reddish brown, flat, oval, and have no hind wings. The front wings are vestigial and reduced to pad-like structures. Bed bugs have segmented abdomens with microscopic hairs that give them a banded appearance. Adults grow to 4-5 mm long and 1.53 mm wide. Feeding habits. Newly hatched nymphs are translucent, lighter in color, and become browner as they molt and reach maturity. A bed bug nymph of any age that has just consumed a blood meal has a bright red, translucent abdomen, fading to brown over the next several hours, and to opaque black within 2 days as the insect digests its meal. Bed bugs may be mistaken for other insects, such as book lice, small cockroaches, or carpet beetles. However, when warm and active, their movements are more ant-like and, like most other true bugs, they emit a characteristic disagreeable odor when crushed. Bed bugs use pheromones and charomones to communicate regarding nesting locations, feeding, and reproduction. The lifespan of bed bugs varies by species and is also dependent on feeding. Feeding physiology. Bed bugs can survive a wide range of temperatures and atmospheric compositions. Below 16.1 degrees Celsius, adults enter semi-hibernation and can survive longer. They can survive for at least 5 days at 10 degrees C, but die after 15 minutes of exposure to 32 degrees C. Common commercial and residential freezers reach temperatures low enough to kill most life stages of bed bug, with 95% mortality after 3 days at 12 degrees C. They show high desiccation tolerance, surviving low humidity in a 35–40 degrees C range even with loss of 1/3 of body weight. Earlier life stages are more susceptible to drying out than later ones. Reproduction. The thermal death point for C. lectularius is 45 degrees C. All stages of life are killed by 7 minutes of exposure to 46 degrees Celsius. Bed bugs apparently cannot survive high concentrations of carbon dioxide for very long. Exposure to nearly pure nitrogen atmospheres, however, appears to have relatively little effect even after 72 hours. Sperm protection. Bed bugs are obligatory hematophagous insects. Most species feed on humans only when other prey are unavailable. They obtain all the additional moisture they need from water vapor in the surrounding air. Bed bugs are attracted to their hosts primarily by carbon dioxide, secondarily by warmth, and also by certain chemicals. Bed bugs prefer exposed skin, preferably the face, neck, and arms of a sleeping person. Bed bugs have mouth parts that saw through the skin, and inject saliva with anticoagulants and painkillers. Sensitivity of humans varies from extreme allergic reaction to no reaction at all. The bite usually produces a swelling with no red spot, but when many bugs feed on a small area, reddish spots may appear after the swelling subsides. Sperm and seminal fluid allocation. Although under certain cool conditions adult bed bugs can live for over a year without feeding, under typically warm conditions they try to feed at 5 to 10 day intervals, and adults can survive for about 5 months without food. Younger instars cannot survive nearly as long, though even the vulnerable newly hatched first instars can survive for weeks without taking a blood meal. At the 57th Annual Meeting of the Entomological Society of America in 2009, newer generations of pesticide-resistant bed bugs in Virginia were reported to survive only 2 months without feeding. DNA from human blood meals can be recovered from bed bugs for up to 90 days, which means they can be used for forensic purposes in identifying on whom the bed bugs have fed. A bed bug pierces the skin of its host with a stylet fascicle, rostrum, or beak. The rostrum is composed of the maxillae and mandibles, which have been modified into elongated shapes from a basic, ancestral style. The right and left maxillary stylets are connected at their midline and a section at the center line forms a large food canal and a smaller salivary canal. The entire maxillary and mandibular bundle penetrates the skin. The tips of the right and left maxillary stylets are not the same, the right is hook-like and curved, and the left is straight. The right and left mandibular stylets extend along the outer sides of their respective maxillary stylets and do not reach anywhere near the tip of the fused maxillary stylets. The stylets are retained in a groove in the labium, and during feeding, they are freed from the groove as the jointed labium is bent or folded out of the way, its tip never enters the wound. Egg production. The mandibular stylet tips have small teeth, and through alternately moving these stylets back and forth, the insect cuts a path through tissue for the maxillary bundle to reach an appropriately sized blood vessel. Pressure from the blood vessel itself fills the insect with blood in 3 to 5 minutes. The bug then withdraws the stylet bundle from the feeding position and retracts it back into the labial groove, folds the entire unit back under the head, and returns to its hiding place. It takes between 5 and 10 minutes for a bed bug to become completely engorged with blood. In all, the insect may spend less than 20 minutes in physical contact with its host, and does not try to feed again until it has either completed a molt or, if an adult, has thoroughly digested the meal. Alarm pheromones. North Carolina State University found that bed bugs, in contrast to most other insects, tolerate incest and are able to genetically withstand the effects of inbreeding quite well. Male bed bugs sometimes attempt to mate with other males and pierce their abdomens. This behavior occurs because sexual attraction in bed bugs is based primarily on size, and males mount any freshly fed partner regardless of sex. All bed bugs mate by traumatic insemination. Female bed bugs possess a reproductive tract that functions during oviposition, but the male does not use this tract for sperm insemination. Instead, the male pierces the female's abdomen with his hypodermic penis and ejaculates into the body cavity. In all bed bug species except Primicimex cavernus, sperm are injected into the mesospermallage, a component of the spermallage, a secondary genital structure that reduces the wounding and immunological costs of traumatic insemination. Injected sperm travel via the hemolymph to sperm storage structures called seminal conceptacles, with fertilization eventually taking place at the ovaries. The bed bug alarm pheromone consists of 2-octenyl and 2-hexanyl. It is released when a bed bug is disturbed, as during an attack by a predator. A 2009 study demonstrated the alarm pheromone is also released by male bed bugs to repel other males that attempt to mate with them. Cimex lectularius and C. hemipterus mate with each other given the opportunity, but the eggs then produced are usually sterile. In a 1988 study, one of 479 eggs was fertile and resulted in a hybrid, Cimex hemipterus times lectularius. Life stages. Sexual dimorphism. Host searching. Aggregation and dispersal behavior. Cimex lectularius males have environmental microbes on their genitals. These microbes damage sperm cells, leaving them unable to fertilize female gametes. Due to these dangerous microbes, males have evolved antimicrobial ejaculate substances that prevent sperm damage. When the microbes contact sperm or the male genitals, the bed bug releases antimicrobial substances. Many species of these microbes live in the bodies of females after mating. The microbes can cause infections in the females. It has been suggested that females receive benefit from the ejaculate. Though the benefit is not direct, females are able to produce more eggs than optimum, increasing the amount of the female's genes in the gene pool. In organisms, sexual selection extends past differential reproduction to affect sperm composition, sperm competition, and ejaculate size. Males of C. lectularius allocate 12% of their sperm and 19% of their seminal fluid per mating. Due to these findings, Reinhardt et al. Proposed that multiple mating is limited by seminal fluid and not sperm. After measuring ejaculate volume, mating rate, and estimating sperm density, Reinhardt et al. Showed that mating could be limited by seminal fluid. Despite these advances, the cost difference between ejaculate dose dependence and mating frequency dependence have not been explored. Males fertilize females only by traumatic insemination into the structure called the ectospermallage. On fertilization, the female's ovaries finish developing, which suggests that sperm plays a role other than fertilizing the egg. Fertilization also allows for egg production through the corpus allodum. Sperm remains viable in a female's spermatheca, a sperm-carrying sac, for a long period of time as long as body temperature is optimum. The female lays fertilized eggs until she depletes the sperm found in her conceptacle. After the depletion of sperm, she lays a few sterile eggs. The number of eggs a Coelectolarius female produces does not depend on the sperm she harbors, but on the female's nutritional level. In Coelectolarius, males sometimes mount other males because male sexual interest is directed at any recently fed individual regardless of their sex, but unfed females may also be mounted. Traumatic insemination is the only way for copulation to occur in bed bugs. Females have evolved the spermalage to protect themselves from wounding and infection. Because males lack this organ, traumatic insemination could leave them badly injured. For this reason, males have evolved alarm pheromones to signal their sex to other males. If a male Coelectolarius mounts another male, the mounted male releases the pheromone signal and the male on top stops before insemination. Females are capable of producing alarm pheromones to avoid multiple mating, but they generally do not do so. Two reasons are proposed as to why females do not release alarm pheromones to protect themselves. First, alarm pheromone production is costly. Due to egg production, females may refrain from spending additional energy on alarm pheromones. The second proposed reason is that releasing the alarm pheromone reduces the benefits associated with multiple mating. Benefits of multiple mating include material benefits, better quality nourishment or more nourishment, genetic benefits including increased fitness of offspring, and finally, the cost of resistance may be higher than the benefit of consent, which appears the case in Coelectolarius. Bed bugs have 5 immature nymph life stages and a final sexually mature adult stage. They shed their skins through ecdysis at each stage, discarding their outer exoskeleton, which is somewhat clear, empty exoskeletons of the bugs themselves. Bed bugs must molt 6 times before becoming fertile adults, and must consume at least 1 blood meal to complete each molt. Each of the immature stages lasts about a week, depending on temperature and the availability of food, and the complete life cycle can be completed in as little as 2 months. Fertilized females with enough food lay 3 to 4 eggs each day continually until the end of their lifespans, possibly generating as many as 500 eggs in this time. Genetic analysis has shown that a single pregnant bed bug, possibly a single survivor of eradication, can be responsible for an entire infestation over a matter of weeks, rapidly producing generations of offspring. Detection. Slide of Coelectolarius. On the right is recently sloughed skin from its nymph stage. Nymph feeding on host. Management. Predators. Epidemiology. Blood-fed stage. Sexual dimorphism occurs in Coelectolarius, with the females larger in size than the males on average. The abdomens of the sexes differ in that the males appear to have pointed abdomens, which are actually their copulatory organs, while females have more rounded abdomens. Since males are attracted to large body size, any bed bug with a recent blood meal can be seen as a potential mate. However, males will mount unfed, flat females on occasion. The female is able to curl her abdomen forward and underneath toward the head to not mate. Males are generally unable to discriminate between the sexes until after mounting, but before inseminating. C. lectularius only feeds every 5 to 7 days, which suggests that it does not spend the majority of its life searching for a host. When a bed bug is starved, it leaves its shelter and searches for a host. If it successfully feeds, it returns to its shelter. If it does not feed, it continues to search for a host. After searching, regardless of whether or not it has eaten, the bed bug returns to the shelter to aggregate before the photophase. Race argues that 2 reasons explain why C. lectularius would return to its shelter and aggregate after feeding. One is to find a mate and the other is to find shelter to avoid getting smashed after eating. C. lectularius aggregates under all life stages and mating conditions. Bed bugs may choose to aggregate because of predation, resistance to desiccation, and more opportunities to find a mate. Airborne pheromones are responsible for aggregations. Another source of aggregation could be the recognition of other C. lectularius bugs through mechanoreceptors located on their antennae. Aggregations are formed and disbanded based on the associated cost and benefits. Females are more often found separate from the aggregation than males. Females are more likely to expand the population range and find new sites. Active female dispersal can account for treatment failures. Males, when found in areas with few females, abandon an aggregation to find a new mate. The males excrete an aggregation pheromone into the air that attracts virgin females and arrests other males. Bed bugs can exist singly, but tend to congregate once established. Though strictly parasitic, they spend only a tiny fraction of their life cycles physically attached to hosts. Once a bed bug finishes feeding, it relocates to a place close to a known host, commonly in or near beds or couches in clusters of adults, juveniles, and eggs, which entomologists call harborage areas or simply harborages, to which the insect returns after future feedings by following chemical trails. These places can vary greatly in format, including luggage, inside of vehicles, within furniture, amongst bedside clutter, even inside electrical sockets and nearby laptop computers. Bed bugs may also nest near animals that have nested within a dwelling, such as bats, birds, or rodents. They are also capable of surviving on domestic cats and dogs, though humans are the preferred host of C. lectularius. History. Bed bugs can also be detected by their characteristic smell of rotting raspberries. Bed bug detection dogs are trained to pinpoint infestations, with a possible accuracy rate between 11% and 83%. Homemade detectors have been developed. Bed bugs are extremely difficult to get rid of. This frequently requires a combination of non-pesticide approaches and the use of insecticides. Mechanical approaches, such as vacuuming up the insects and heat treating or wrapping mattresses, are effective. An hour at a temperature of 45 degrees Celsius or over, or 2 hours at less than 17 degrees C kills them. This may include a domestic clothes dryer for fabric or a commercial steamer. Bed bugs and their eggs will die on contact when exposed to surface temperatures above 180 degrees, and a steamer can reach well above 230 degrees. A study found 100% mortality rates for bed bugs exposed to temperatures greater than 50 degrees Celsius for more than 2 minutes. The study recommended maintaining temperatures of above 48 degrees Celsius for more than 20 minutes to effectively kill all life stages of bed bugs, and because in practice treatment times of 6 to 8 hours are used to account for cracks and indoor clutter. This method is expensive and has caused fires. Starving them is not effective as they can survive without eating for 100 to 300 days, depending on temperature. For public health reasons, individuals are encouraged to call a professional pest control service to eradicate bed bugs in a home, rather than attempting to do it themselves, particularly if they live in a multifamily building. As of 2012, no truly effective insecticides are available. Insecticides that have historically been found effective include pyrethroids, dichlorvos, and malathion. Resistance to pesticides has increased significantly over time, and harm to health from their use is of concern. The carbamate insecticide Propokar is highly toxic to bed bugs, but it has potential toxicity to children exposed to it, and the U.S. Environmental Protection Agency has been reluctant to approve it for indoor use. Boric acid, occasionally applied as a safe indoor insecticide, is not effective against bed bugs because they do not groom. The fungus Bovaria bassiana is being researched as of 2012 for its ability to control bed bugs. As bed bugs continue to adapt pesticide resistance, researchers have examined the insect's genome to see how its adaptations develop and to look for potential vulnerabilities that can be exploited in its growth and development phases. Natural enemies of bed bugs include the masked hunter insect, cockroaches, ants, spiders, mites, and centipedes. However, biological pest control is not considered practical for eliminating bed bugs from human dwellings. Bed bugs occur around the world. Rates of infestations in developed countries, while decreasing from the 1930s to the 1980s, have increased dramatically since the 1980s. Previously, they were common in the developing world, but rare in the developed world. The increase in the developed world may have been caused by increased international travel, resistance to insecticides, and the use of new pest control methods that do not affect bed bugs. The exact causes of this resurgence remain unclear. It is variously ascribed to greater foreign travel, increased immigration from the developing world to the developed world, more frequent exchange of second-hand furnishings among homes, a greater focus on control of other pests, resulting in neglect of bed bug countermeasures, and increasing resistance to pesticides. Declines in household cockroach populations that have resulted from the use of insecticides effective against this major bed bug predator have aided the bed bug's resurgence, as have bans on DDT and other potent pesticides. The fall in bed bug populations after the 1930s in the developed world is believed partly due to the use of DDT to kill cockroaches. The invention of the vacuum cleaner and simplification of furniture design may have also played a role. Others believe it might simply be the cyclical nature of the organism. The common bed bug is the species best adapted to human environments. It is found in temperate climates throughout the world. Other species include Cymex hemipterus, found in tropical regions, which also infests poultry and bats, and Leptocymex boetti, found in the tropics of West Africa and South America, which infests bats and humans. Cymex pilocellus and Cymex pipistrella primarily infest bats, while Haematosiphon inodora, a species of North America, primarily infests poultry. In November 2016, a media report noted that tropical bed bugs, Cymex hemipterus, which had been extirpated from the state during World War II, were discovered in Brevard County, Florida, and are expected to spread in distribution within the United States. 20th century resurgence. Coelectolarius may have originated in the Middle East in caves inhabited by bats and humans. Bed bugs were mentioned in ancient Greece as early as 400 BC, and were later mentioned by Aristotle. Pliny's Natural History, first published circa AD 77 in Rome, claimed bed bugs had medicinal value in treating ailments such as snake bites and ear infections. Society and culture. Bed bugs were first mentioned in Germany in the 11th century, in France in the 13th century, and in England in 1583, though they remained rare in England until 1670. Some in the 18th century believed bed bugs had been brought to London with supplies of wood to rebuild the city after the Great Fire of London. Giovanni Antonio Scopoli noted their presence in Carniola in the 18th century. Traditional methods of repelling and/or killing bed bugs include the use of plants, fungi, and insects, such as black pepper, black cohosh, Pseudothria hookerii, Lagra alida, Eucalyptus saligna oil, henna, infused oil of Melilantha vulgaris, fly agaric, Actia species, tobacco, heated oil of terebinthina, wild mint, narrow-leaved pepperwort, Myrica species, Robert geranium, bugbane, herb and seeds of cannabis, Opulus berries, masked hunter bugs, and many others. In the mid-19th century, smoke from peat fires was recommended as an indoor domestic fumigant against bed bugs. Dusts have been used to ward off insects from grain storage for centuries, including plant ash, lime, dolomite, certain types of soil, and diatomaceous earth or kieselguhr. Of these, diatomaceous earth in particular has seen a revival as a non-toxic residual pesticide for bed bug abatement. While diatomaceous earth performed poorly, silica gel may be effective. Basketwork panels were put around beds and shaken out in the morning in the UK and in France in the 19th century. Scattering leaves of plants with microscopic hooked hairs around a bed at night, then sweeping them up in the morning and burning them, was a technique reportedly used in southern Rhodesia and in the Balkans. Bean leaves have been used historically to trap bed bugs in houses in Eastern Europe. The trichomes on the bean leaves capture the insects by impaling the feet of the insects. The leaves are then destroyed. Prior to the mid-20th century, bed bugs were very common. According to a report by the UK Ministry of Health, in 1933, all the houses in many areas had some degree of bed bug infestation. The increase in bed bug populations in the early 20th century has been attributed to the advent of electric heating, which allowed bed bugs to thrive year-round instead of only in warm weather. Bed bugs were a serious problem at U.S. military bases during World War II. Initially, the problem was solved by fumigation, using Zyklon discoids that released hydrogen cyanide gas, a rather dangerous procedure. Later, DDT was used to good effect. The decline of bed bug populations in the 20th century is often credited to potent pesticides that had not previously been widely available. Other contributing factors that are less frequently mentioned in news reports are increased public awareness and slum clearance programs that combined pesticide use with steam disinfection, relocation of slum dwellers to new housing, and in some cases also follow-up inspections for several months after relocated tenants moved into their new housing. Bed bug infestations have resurged since the 1980s for reasons that are not clear, but contributing factors may be complacency, increased resistance, bans on pesticides, and increased international travel. The U.S. National Pest Management Association reported a 71% increase in bed bug calls between 2000 and 2005. The number of reported incidents in New York City alone rose from 500 in 2004 to 10,000 in 2009. In 2013, Chicago was listed as the #1 city in the United States with the worst bed bug infestation. As a result, the Chicago City Council passed a bed bug control ordinance to limit their spread. Additionally, bed bugs are reaching places in which they never established before, such as southern South America. One recent theory about bed bug reappearance in the U.S. is that they never truly disappeared, but may have been forced to alternative hosts. Consistent with this is the finding that bed bug DNA shows no evidence of an evolutionary bottleneck. Furthermore, investigators have found high populations of bed bugs at poultry facilities in Arkansas. Poultry workers at these facilities may be spreading bed bugs, unknowingly carrying them to their places of residence and elsewhere after leaving work.